It is the duty of leaders to lead, of the creative to create, of the daring to do. The free world expects leadership of us. Its fate and our fate depends upon our leadership. We are industrious, inventive, restless, with the fires that burn within us. But I say that nothing is easy, and the best things are the hardest. And all our troubles, all our immense difficulties, now and in the future, can I say, be solved if we have the will, the courage. The future is to those who take it.
Please join me in welcoming Sue Siegel to the stage, who will then welcome Craig Venter. But Sue?
Thank you, Steve. And I am going to introduce a man that needs no introduction. I suspect most of you know that. If you think back to ESTs, how many of you know what an EST is, actually? Different audience, So Craig, way back when he was at the NIH, essentially found ESTs, which are expression sequence tags. And a lot of people said, you cannot sequence the human genome using ESTs. This was the first sort of incarnation of doing sequencing. And as you will find, a pattern in what Craig has done is essentially proved pretty much everybody wrong, okay? So that was one of those first things. He then went off to found HGS, Human Genome Sciences. He was a co-founder there. He founded Tiger, which, if for those of you who don't know Tiger, different audience, this was the Institute for Genomic Research, and this was the first sequence of a prokaryote, so bugs, essentially was done by Craig, okay? A lot of folks don't know that. And then he went off and he said, now I'm going to start Celera. He had this vision that, in fact, if you took a bunch of machines, you put them all together, and you put the power of company and resources behind it, you could sequence the human genome faster than anybody else could. And in fact, what did he do? You know that the human genome was supposed to be done by the year sort of 2002, 2003. When was it done? 2000. Okay, so he did it. And then people said, well, what is he going to do next? And remember, during that time, it was also amassing all of the information you could possibly imagine and created Celera, and this model of informatics and data is going to be the currency of the future. After that, if that wasn't enough, sequencing the human genome, come on, right? Okay, so we do it now every day for a little bit of time, and it's a lot of less money. Back then, man, it was really hard. So first one. And then he said, now I'm actually going to go, and I am going to figure out sort of the next thing. He created the JCVI Institute, and he said he was going to go comb the oceans and see and sample the oceans to see what in fact might be found, because we really didn't understand that uncharted territory. So human genome, uncharted territory, ocean, uncharted territory in many ways. And he went and sampled, and he came back, and he sequenced millions and millions of organisms, of which he discovered a number of them, when you start to think about that. And so he did it again. And during that time, he also created synthetic genomics. Are you guys tired yet? Because this has happened in like 20 years. He created synthetic genomics, and what did he do? He synthesized the first organism. Okay, and we're going to explore that a little bit. So again, Craig, not having done enough, said, well, now I'm actually going to bring together all the things I've been doing before, but I want to do something about translation of it too. And I'm going to create Human Longevity Institute. And that's what he set out to do. And again, we're going to talk a little bit about that today. So with that, I'm going to welcome Craig on stage so that we can actually talk a little bit about what he does in his spare time, which I'm about to tell you what he tells in his spare time, which he wouldn't tell me, but I got it from my little sources out there. His favorite TV show, The World of Dumbest Criminals.
That's not my favorite.
Okay, his favorite is Jay Leno's Garage. And chasing classic cars and American pickers. There's a theme, you guys, right? And as you might imagine, he's got some nice cars. All right, Craig. here we go.
I'm happy to sit over here and let you keep talking about it.
No, enough of that. Now we're going to get to some of the hard stuff. So first of all, tell us about Human Longevity Institute. What are you doing? What are you thinking? Where are you going?
It's Human Longevity, Inc. So people confuse
Sorry.
these things because the Venture Institute's a not-for-profit institute. Human Longevity is definitely not-for-profit. It's not yet profitable, but it's not intended to be a not-for-profit. So the goal was, when we finished the first genome, it was hard to interpret. In fact, that hasn't changed much in 15 years. And we realized we needed really large numbers of genomes, and we needed phenotype information to go with them. So we've set out to try and do 1 million genomes by 2020 and collect phenotype information on everybody whose genome we're sequencing. So it's a huge informatics challenge. It's a computational challenge. And we're adding into it machine learning to try and put this data together. So it's mixing all these together. Just in our startup phase, we passed 20,000 human genomes, all with phenotype information, and using GE and some other equipment of making some combination findings with the genome that I think are going to blow people away.
That's awesome. And by the way, you can't do it unless you use GE instruments, just FYI. Just kidding. In reality, though, you talk about, and again, this is a different audience than what you typically speak
If
to.
they didn't know what an EST was. So it's
Well, and so that's why I'm going to say, I want to make sure that everybody understands, the notion of genotype and phenotype, and explain what you're trying to do as you bring together many different phenotypes and not just genotype, and how important that is.
So the way discoveries have been made before most of you entered science was you had to get very large pedigrees of people with common ailments and get a little bit of information to try and map those genes. So things like the Huntington's gene took about 25 years and maybe 10 major research groups around the world to find. Now having phenotype information and really accurate genome information, we can make those same discoveries on a single person. And the correlation between getting an estimate of a risk from the genome is totally different when you have a yes-no answer. Do they have this trait? Do they have this disease? So what it makes, the data builds on each other, so it makes the predictions much more accurate and from mere statistics. And for example, BRCA1 and BRCA2 aren't actually clearly associated with the cause of breast and ovarian cancer. They're markers that get us in the vicinity.
Okay.
And so it's only a 50-50 chance of getting breast or ovarian cancer if you have changes in both BRCA1 and BRCA2. The thing that pushes it into meaningful territory is if you have a family history of breast and ovarian cancer, where basically every woman in the family has breast or ovarian cancer. That means there's other genetic elements that are actually causing the cancers that BRCA1, BRCA2 get us in the vicinity of knowing. So that gene information coupled with the history is very predictive. The gene information on its own for BRCA1 and 2 is only 50-50. So we have to improve the accuracy and get to the things that actually cause the disease versus are just kind of in the neighborhood.
So contextual information is going to be really important, and how you're adding that into the equation. So particularly for the digital health audience, how are you thinking about those kind of technologies coming in to become another phenotype as you gather all this data?
No, in fact, the digital world is a challenge, and so in 1999, we had to build the third largest computer just for assembling the first genome, and that cost about $50 million and was only 1.5 teraflops. So
And a teraflop is 10 to the...
it's $100 today, if that puts it in context.
All right, got it.
Okay, so there's been a slight change, and with cloud computing, we can use distributed computing and not have to worry about building a machine to do this. Although when you do a brain MRI image, it generates about 3 gigs of data. And a group of scientists, computational scientists at UCSD, led by Anders Dale, developed an algorithm that takes that 3 gigs of data and converts it into a single page, a table of volumes of different brain regions, where they can detect the slightest changes in repeated MRIs over about 3 months that can detect whether you're developing dementia or not. So that information coupled with the genome is very powerful, but it takes 3 gigs of data and converts it into a few KB of data that then is easy to take into a machine learning algorithm with lots of other cohorts of it. So it's not just the sheer volume of data, it's the intelligent reduction of the complexity so it's usable for comparing to everything else.
And tell us a little bit about what you're doing with regards to Franz and face recognition. Tell the story.
Yeah. So Franz Ock was hired out of Google into HLI. I'm sure you know what he developed. He developed Google Translate. So if you haven't used that, I'm definitely in the wrong room. And he used a unique machine learning approach to convert languages into each other. So I convinced him the human genome was the ultimate translation problem. Moonshots are too nearsighted. I think
Wow.
You guys need to aim a little bit higher and further. Anybody can get to the moon these days.
So what's yours, just so we sort of put it in position here?
Well, I think the next decade in planetary space, obviously colonizing Mars is where people are headed, and things like being able to send biology through the internet, like my institute and synthetic genomics developed, are ways to get things to and from distant planets. But I think that the challenges we've been talking about in this space, you have to think well ahead of where the crowd is going.
Facial recognition.
Yeah.
Go to face recognition for a sec.
So using machine learning, it's a way of doing what thousands or even tens of thousands of scientists themselves can never get to with looking at a small bit at a time. So the first challenge I gave to Franz and his group was to see if we could predict your photograph straight from your genome, just A's, C's, G's, and T's. And we'll be submitting a paper on this shortly.
I was much prettier, by the way. They did it, and they came up with a picture much prettier than what I am, seriously.
Well, it predicts your age just post-puberty. You might wonder what your genome codes for. It codes for what you look like post-puberty. And the algorithm does a little smoothing, so it does make you look good. You're okay on your own, but we all could use a little smoothing.
Open mouth, insert thigh. Very good, Craig. That was good.
And the algorithm also makes faces totally symmetrical right now, which they're not. But we're very good at predicting your face. But if we're recording your voice just from these microphones, we can accurately predict your sex, your age, and your height. So there's information contained in everything that's human. And it's a matter of using new approaches to pull this out. So right from your genetic code, I was absolutely wrong. I said you would get your genome sequenced once in life unless you had cancer. It turns out the exact age somebody is when they have a blood sample drawn is essential, because we can now predict your age right from your genetic code. It changes throughout life. It becomes a measurement of aging. So using machine learning, we compiled everything that was known about human traits and physical predictions, and the machine learning out-predicted those by a substantial margin. So just knowing the components wasn't sufficient. And the other thing machine learning does is what your body does. It uses information across the genome. It doesn't just use one gene and one SNP in a gene like we've been measuring. It uses this information in an integrated fashion, and that's key to understanding different diseases, complex diseases, understanding aging, et cetera.
Got it. So in 2010, you did an interview with Der Spiegel. And in that interview, you talked about the medical value that had been derived from the genome in 2010 was close to zero. Yeah. So we're in 2016. Tell us a little bit about, are you changing your mind on that? And then tell us about 2025.
So It's moved more in the last 2 years than it's moved in the previous
Why?
13 years.
Okay, why?
Well, for these same reasons. We had small data sets, nothing to compare to each other. My genome was the first one that was done on the internet, but there's less than 1,000 actual genomes that have been completely sequenced that are out there.
What about, that are out there? Because I was going
Well-
to say, what about the Manchester Project in the UK? I would have
Well,
thought.
so nomenclature here is important.
Okay.
Genome has become a very loose word. Back when we were first doing genomes, it meant you actually sequenced every base pair and closed every gap. That's not possible with the human genome. Sequencing covers only 85% of the genome. But there was a paper just published in Nature said the sequence of 1,000 human genomes, but they were only done at an average of 7 to 8x coverage. So that may sound impressive if you know nothing about mathematics of genomics, but it means less than a third of the genome was covered. So we sequence things to 30 to 40x at HLI, and we have incredible accuracy, and I'll show some of this tomorrow morning, where we sequence every base pair on average at least 10 times and have a low error rate and a low false positive, false negative rate. But even with that, only 85% of the genome is covered. So when somebody's saying they're sequencing a genome, it's important to say at what depth and what technology.
And you know, you're now using this in a program called Nucleus, in your Nucleus Health program, right? So tell us a little bit about what that is.
So the Health Nucleus is our ultimate phenotyping center. So we have the latest GE 3T MRI machine, and we have protocols that only we in GE Research have. We only have, we have DEXA scanners. We're adding CT and a second MRI. We have 4D echo cardiograms. We have ways for measuring neurological functions and phenotyping. And we measure the human genome, the microbiome. We bleed people and take 18 tubes of blood and measure literally
You're supposed to be selling
thousands
this,
of chemicals. I'm sorry. Well, we don't do it from spit, so it doesn't quite work that way. And we integrate this information. So just the imaging has been absolutely stunning in terms of the discoveries. The 4D echo, we've discovered 2 people with aortic aneurysms that thought they were totally healthy. And we integrate this data, and you find a physical trait, and we can go into the genome, and we found a gene duplication event associated with the aortic aneurysms. We can find different types of brain tumors and find the genes associated with them. So it's not hypothetical anymore. We can start with the genome and go the other way, and we predict these people might have these diseases because they have a higher risk, and we score it. They do or they don't. We can detect whether they have cancer. We can detect whether they have enlarged hearts, whether they have different types of defects in the brain, whether they have dementia. So it's intense phenotyping along with an incredibly accurate genome that allows us to put the data together in a combination that's going to be, I think, really stunning when people see all the outcome.
And who actually meets with the consumer? Because it's not a patient anymore. It's really a consumer who is choosing to do this, right?
Yeah.
Who meets and actually discusses what you're finding? Because the information complexity is huge.
Yeah. So we've developed a new approach. We actually make 3D avatars with the, I think there's 400 cameras in the system. Once you make an avatar, you guys have all seen the movies, you can digitally make them do whatever you want. When I'm not there, they make mine do a funny dance of some sort. And your avatar will walk you through all this data. So we start with your avatar. We start with the actual predictions of what you look like. It walks you through the MRI data all the way down to your skeleton with the DEXA, down into your genome, and goes through this data. But the data goes through and is presented with physicians and genetic counselors. It's too much complex information. And we only have healthy people come in.
Or so they think.
But they don't leave healthy. So either we're doing something really unique to them while they're there. But that's just it. The statistics, this is kind of a younger group, but if you're between 50 and 75 and you're a male, you have a 30% chance of dying in that time period. And a third of that is from cancer and another third is from heart disease. People just don't know they have heart disease or cancer until they get to the symptomatic stage. If you're a female, it goes down to a 20% chance of dying, but still a third is from cancer and a third is from heart disease. 1.3 million people get diagnosed with cancer each year. Those cancers just didn't appear a few minutes before they were diagnosed. So a third of the population is walking around with cancer, with heart disease, with pre-dementia symptoms that they're unaware of. The difference is when we find them early, they're treatable or preventable or switched into a combination of the two. The problem we have with this comprehensive cancer work we're doing, we get individuals when they're discovered and diagnosed with cancer at stage 4, where the odds of successful treatment are way down versus what we have. So I'll talk tomorrow about one case, a tumor discovered under the breastbone that showed up with the MRI imaging, was removed. It hadn't penetrated the tissue, and the guy went home 2 days later, completely healthy, cancer-free, and risk-free. Had he not had this test, probably in a few years it would have penetrated the tissue, and the survival time goes down to about a year.
Wow. So medical value of what?
Medical value, but the predictive value is what we're building out of this database. So in the future, as you get your genome sequenced, these things will be predictable right from the genetic code, the same that your face and these other physical traits
But
are.
aren't you a little worried about what that's going to do between CRISPR-Cas9 and what you're able to do in terms of what we might be able to plan as it relates to the kind of populations we want? How do you think about that?
Well, I think right now, and I've written about this, I think that editing the human genome is inevitable, but I think we should wait.
Say that again?
It's inevitable that we will do it, but I think we need to wait till our knowledge base is a lot better. And I liken back to discoveries that were made early on in Drosophila. There was early on a developmental gene that helped form the limbs and tissues, that in the late stage that same protein became a structural protein in the wings of the fruit fly. We think we know a function for a gene, and if you go modifying it, you don't know what functions you're changing. So we can't afford to do it with human experimentation without having clear-cut knowledge. That being said, there are some diseases, maybe like sickle cell anemia or ataxia, telangiectasia, that are reasonable and ethical to change.
That you'd say go ahead.
I would do it if it was my family.
So it's single gene, well understood.
And devastating diseases where the outcome is clearly understood. The challenge for everybody as we start to predict traits, and right now with circulating fetal cells in the maternal bloodstream, we can sequence the genome and we can give the families a picture of their hypothetical child at age 18.
And they might not like what they look like.
They might not like what they look like. They might find it was the wrong father. There's a lot of things people could find. There's surprises that come up later. When you see them in advance, it might cause some increased legal action. But it's a challenge as we learn how to read the genetic code. It is our software of life. We're learning how to read the software. At the Venture Institute and at Synthetic Genomics, they're learning how to write the software. With CRISPRs, everybody can edit the software. But we're very early on in our knowledge curve. 10 years ago, we knew virtually nothing. We're just starting to get there.
Yep.
We're at the earliest stages with cancer, but we're learning rapidly at least some ways to alter the outcomes.
So you synthesized an organism. And this was 2 years ago, 3 years ago?
5 years ago.
5 years ago.
Time flies.
So what? Now what's happened? Okay, what have you done since then, and what should we be thinking about as you've continued to progress that?
So with the Venture Institute, the not-for-profit institute, and another for-profit company, Synthetic Genomics, that's presenting at this conference as well, over the last 5 years. So the first one we did was sort of basically copying most of a known species genome and seeing if we could actually reproduce it from 4 bottles of chemicals. And we were able to with some key alterations. Then we set out to design a new species for the first time, and we thought it would be pretty straightforward to do this on first principles. First principles of biology don't get you to a living cell. So the reason it's taken us 5 years to do is there were a lot of iterations of things, testing them to see if they worked and didn't work. And sort of a story that goes along with this, when I was on a book tour of my book about synthetic life at the speed of light, I was in Seattle, and my late uncle was one of the lead designers for the Boeing 757. And I said, imagine building a new aircraft without knowing what all the parts do. And he shouted from the back of the room, "What makes you think we knew?" So designing life is kind of like building aircraft. If they fall out of the sky, they find out what's wrong and they fix it on the next iteration. We kept testing until we found what was needed. And so that paper hopefully will be out soon, describing the first species designed through this combination of actual design and iterative testing to get something that would work.
Wow, and when you say that would work, so it has functional life.
Well, it lives and self-replicates. It's not extremely sophisticated, but
So it lives and
as
it has
evolution
sex.
goes,
That's kind of cool, right? Just over and over again. That's kind of cool.
Bacteria don't have sex.
Oh, bacteria don't have to. Sorry. Okay. All right. So, in all frankness, I mean, how do you measure that in fact it's functional? What was proving that in fact it's real?
It can make billions of copies of itself.
And they're
if
all
you
identical.
could do that, people would be very impressed.
Okay. So when is this coming out?
Hopefully in a month or so.
And
It's just
how
on the final review stages in a scientific journal.
how complex is this organism?
It has the smallest genome of any self-replicating organism ever known. We tried to get to what a minimal set would be. Yet its software is still immensely complex. It has all these regulatory functions. It has what people consider epigenetics. And the point is, all of that is coded for in the linear code of the DNA. And I think that's a surprise to a lot of people. Because you can get a separate class on epigenetics, you think it's a separate phenomenon. All that is coded for in that linear basic code that we all have as our software. So it doesn't matter whether it's minimal cell or a human cells, that's why understanding the software is so critical.
So if that took 5 years, and you think about exponential technologies, and the speed of all the convergence of the technologies that are coming together, what's your Martian shot against that?
I guess the right thing would be creating life in 7 days.
I did ask for that, didn't I? I mean, seriously. Because it's just mind-boggling when you think about what you actually can do.
Or 6 days to improve upon history.
So there's no rest. There's absolutely no rest. So, all right, we're going to wind down now, and I'm going to shift gears and just really ask you this. You've got a room full of entrepreneurs and folks that want to create the new. And as you think about all the things that everybody said you can't do, he'll never do it, and then you just did, what advice would you give to some of the entrepreneurs in here? What would you say in terms of how they go about thinking of their own futures and what they should go do?
I think it's essential to have a belief in what you're doing, and it can't be a delusionary belief, or you'll
But
get.
people thought you were delusionary.
Well, and that's
I
what.
mean, that's the reality. He's like, what? He's going to create synthetic. I mean, what?
And had we failed, that's how it would have gone down in history. So That's how you build confidence in your own predictions, in your own intuition, by actually doing things. If you're right, it's like making movies. You make a blockbuster movie, somebody will give you money to make the next movie. After you've made 10 blockbuster movies, you can have a few failures and somebody will still give you money to make another one. But if you never have a success, you can't really be a successful entrepreneur. So
But some of the entrepreneurs are starting from scratch. This is their first
yeah.
time.
Well, you have to pick your battles. You have to really be passionate about it. You have to believe in what you're trying to do. And you have to pick the right thing. It's very easy in science to pick questions to work on. Picking the right questions that can be answered now with a certain technology is the magic of making success in science. It's that easy. If you can pick thousands and
Somebody
thousands
write that
of
down.
questions.
Did somebody write that down? Because that was good. What did you say? Say it again.
No, you have to pick the right question to answer. And if your timing is off, you can be too early or too late. Right now, I think our timing was perfect on starting human longevity. 5 or 10 years from now, it would definitely be too late. And if we did it 5 years earlier, it would have been way too early. So I set a very clear criteria of what kind of computational power there had to be in the world to do what we were doing, and what kind of sequencing technology. And the Illumina X10 just got to the threshold. We can improve on it tremendously, but it set a threshold of what it would take to get high throughput, highly accurate sequence. But the compute side had to be there as well, and new approaches like machine learning had to be there. None of those existed in a realistic way 5 years ago. So it was a narrow threshold where this could just start to happen.
With that, Craig, thank you for all of your pioneering work, for advancing the sciences like you've done. Thanks so much.